So my name's Kim Carter. I work at Minderoo Foundation, so in a large philanthropy here in Australia. We're a global philanthropy, but based in Australia. I work as part of the Cancer Impact Mission in a program called Unlocking Patient Data, and we're seeking to unlock patient data to ensure, to basically facilitate global patient data sharing at an unprecedented level for the benefit of cancer research and for cancer sufferers everywhere. To really set the scene before my esteemed colleagues take stand, I think everybody in the room's been touched by cancer at some point, whether it's directly yourself, whether it's through friends or family. Cancer's a global disease, and really we don't truly know the extent of it. Global numbers that you see in cancer burden are really estimates at best. Even in first world countries, we don't really have a true real picture of what the burden is, and certainly the burden's measured not just in terms of incidence and prevalence and cases. It's really, there's a lifelong burden of disease, suffering and disability and impacts on patients in particular, well beyond if you're one of the lucky ones who survives your treatment. So it's certainly a challenging problem, and as depicted on the right-hand side, you can see there, it's really a global problem. It impacts all nations to a greater or lesser degree. It doesn't matter whether you've got a first world system or not. Cancer is one of the leading causes of early mortality. As a data person, data certainly can change lives and can save lives. I think the small text on there is probably a little bit hard to read, but with the genomics revolution that we've experienced over the last 20 years, the advent of molecular therapies is a really exciting place to be. And if you're one of the lucky patients who has a particular type of mutation that can be targeted, for example, if you've got a BCR-ABL fusion, you might find you have a collection of drugs available to you now. There's 4 or 5 listed in there, and you've got CML or something else. There's fantastic treatment options available for some patients, which is brilliant. And in fact, with multiple treatment options available, it allows you to really harness real-world evidence and conduct systematic studies and really start to determine, what is the best therapy for each patient. On the right-hand side on this plot is from data from the Australian Institute for Health and Welfare, the last sort of 35 odd years worth of cancer survival statistics. One way of looking at whether we're improving cancer care and showing in Australia at least an upward trend in terms of survival, and this is grouping all cancers together. On the whole, we're starting to survive cancer more than we were in the 80s, which is fantastic, and that sort of trend is seen certainly in most of the first world countries in the globe. If you dig a little bit deeper, though, data can certainly save a lot more lives, and brain cancer is a cancer that, as a foundation, we invest heavily in as one that's not as well supported as other cancer types. And certainly this plot here, again from Australian data for the last 35 years, this is in adults, but pediatrics, it's very much the same. So for brain cancers, which is really not one cancer, it's more than 100 different types of cancer, the survival rates really haven't changed in the last 3 decades. We haven't had lots of new drugs developed, and the hard-to-read meta-analysis on the right-hand side there, despite combinations of therapies and some new exciting targets out there, on the whole, they've been largely ineffective, and we're still looking for that silver bullet that suddenly changes patients' lives. There's lots more work to be done, and something else that doesn't get talked about a lot is also the financial implications for patients. So this is some old numbers, but $200,000 US a patient, for your first year of treatment is enormous burden on the patients in terms of what you face, let alone the physical impacts of the treatment you undergo. So what we really want to do, and what we're trying to support as a foundation, is bringing this sort of notion of patient-centric oncology. We want to really bring about this multimodal picture of patient data, telling the entire story of a patient, or the most complete patient journey that we can put together. We want to tell it from the point where patients get diagnosed or earlier, right through their various treatment options, through to whatever the particular outcomes they come. And there's a lot of different sources that we can get that information from. And in particular, one that probably hasn't been talked a lot about so far is patient-contributed data as well. That the patients themselves can actually contribute quite valuable data to the process beyond what we can pull out of clinical systems, out of registers and so on. You're going to hear the word complexity a lot for the rest of the next couple of talks, but just something we wanted to put up in front of you was the complexity of oncology data. So we have a fantastic data model that we've been hearing a lot about today that's mature and enables global federated research at an unprecedented level. When you get into oncology data, things get a little bit more interesting, and some other diseases as well. But when we want to start telling a fairly complicated patient journey where it's not a simple beginning, middle, and end in terms of treatment, where there's backward steps. You start off with a first-line therapy, patient doesn't respond, you suddenly have to go back and try something different. There's backwards and forwards regression, and it's over potentially a number of years. And you suddenly start to find you've got a lot of data points. It's not a very linear process that you're trying to tell with that. And a lot of data models may not represent that type of data very well. So that it's not a singular event or activity doesn't lend itself well to data models. And that's something that we really want to capture. And probably one further example, and this is from liver cancer, which is another disease we work on a lot. It's that one cancer does not equal one thing to measure, that within a single person's liver cancer, you can have 3, 4, 5, 10 different types of cancer that are completely genetically different or genomically different. Each one of them can be treated with a different kind of therapy. They have different kinds of procedure codes associated, where they come with different measures. They're in a different location, and suddenly you start to get to the point where they're almost completely separate pieces of data to be stored. And again, a data model that fits that is there's not a lot out there to fit that. And I know my colleagues have been underway on this journey for a long time, developing a data model that can start to represent this complexity that we want to tell on the patients in a way that's built around the OMOP data model. And on top of that, we want to start to integrate other pieces of information like genomics, like imaging, and other critical pieces of information in the story. So just to jump back one final step of why I'm standing up here. So as being a part of philanthropy, we're really trying to be disruptive, to be an agent of change, to bring in essentially untied dollars rather than standard research funding, to try and act as an agent of change, to say, who's got a great idea? How do you want to work? What are the possibilities that you can do with this data, with this research? How do you want to help patients now rather than 5 years' time when you get that research grant accepted? So we're working in this space trying to really just make things happen. And two of the key pieces we're trying to support are federated research approaches to break down barriers to collaboration. And certainly the Odyssey community is a real leader in this space. And the other piece that we're really trying to support is a global standard for oncology data. So we're really trying to push the envelope through the OMOP team to support this. And I think you'll now hear from Arcia now to tell you a lot more about why this is critical.
You have heard it before, but there are a lot of new faces. I just give you a little bit more about the complexity of the situation and why are we talking about oncology here? Why should we, right? One sentence, cancer is complicated. It's way more complicated than any other diseases, and we just saw from Kim. I'll give you another example to see that. This is a label, an FDA label for capmatinib, a drug for the indication of metastatic non-small cell lung cancer, patients who have skipping mutations in their exon MET exon 14 skipping mutations. That's a lot of information there. We're all here to answer questions, to generate clinical evidence, to understand what these patients are, who these patients are, what are they being treated, what potential side effects or adverse events they're experiencing, how do they benefit from drugs, what's the disease burden, and a lot more, their quality of life, their survivorship, and so on and so forth. Just take a look at this. I know a lot of us work on oncology, and we have these examples of follicular lymphoma, brain cancers, very, very rare, or DLBCL, hereditary leukemia with 80-something patients in the world, the relapsed refractory disease. But lung cancer, the most common cancer, the most common cause of cancer death in the world. These are the numbers that you see in the States. Around over 200,000 cases are diagnosed with the disease. The moment you try to get to that specific patient population that you want to study, like there's a patient has metastases, they have adenocarcinoma, they have a specific mutation that I want to study, all of a sudden, you'll end up with 7,000 new cases, 7,700, right? It's a small number. Think about your database. That 7,000 in your specific institution will be 100 max, 50, if you're lucky, 150. Generating a reliable evidence for that small size is going to be a little bit hard, and we want to know more. Every day we want to know more. Reality is that this is actually even way more complex. You don't need that. And I'm standing here in 5 years, I'm going to promise you that's going to be even worse than that. Because we get to know more, and we want, like, that's, and we have to try to keep up to answer the questions. So the oncology work group was formed, my math is bad in my history, but probably around 2016, with one goal: to provide a foundation for representing cancer data at the level of granularity and abstraction that enables observational cancer research. And with that, there are 3 solutions that the work group has been working on: creating an oncology network or enabling an oncology network, the oncology module, and also working on the terminologies. So the question came earlier this morning, the numbers are low, right? My institutions have 50 patients, one institution is 120. Within a common data model, if everyone has the same standards, and if we work together, we're able to put together a data network and learn more from all the data that we can get from different places to contribute more to patient care, to our understanding of the disease. The second piece of the work that the work group has been working on is the oncology module. This has 3 pieces. How do we represent the disease? Remember, metastatic non-small cell lung cancer with a mutation in exon 14 skipping mutation, that's a lot. In the data, I see lung cancer. So what we do is we've developed conventions to represent that, represent cancer treatments. You saw on Kim's slide, cancer treatments are not one individual intervention or administration of drug A or drug B. Cancer treatments are usually administered in a combination of drugs with specific dosing and scheduling. That's what we want to see, whether my patients is benefiting from this combination or the other combinations. What are the adverse events they're experiencing? And finally, the episode model. The disease is basically, you can describe it as specific, or periods, continuous periods of time that have specific meaning. A metastatic disease has a different meaning than a localized disease. It's an episode of progression when a disease is actually getting worse has to be specified. Same with treatment. Remember, we're thinking about a combination of the treatment with specific schedule and dosing and timing. We would like and we need to specify that so that we can accurately and reliably study cancer. Just a very quick overview in terms of disease representation. We represent disease as a combination of disease topology and histology. So adenocarcinoma that occurs in lung, plus a set of attributes that makes it specific to your patient population. The patient has metastases, they have mutations in this specific genes. The tumor has a specific dimension. Whatever your patient population is, this combination would allow you to get that. In addition to that, you can also look into this combination of treatment, which we're familiar with, treatment regimens, lines of therapy, the cycles, the doses of cycles, and so on and so forth, and represent them in the episode. And finally, the episodes, right? So continuous periods of the disease or treatment that have distinct meaning, and there is a way that it's essential to describe, to use it for oncology research. I'm not going to go through this, but if there's an interest, tomorrow there's a workshop. We're happy to talk about that. So putting back again the picture that Kim just showed, the model allows us to describe the patient from the time that they're diagnosed with lung cancer till the end of death or end of follow-up as lung cancer. I can identify the period of the time that the patient had metastatic disease. That's an episode of the disease. And the episodes, distinct episodes of progression in the patient's history, also the treatment episodes. Having that picture, there's a lot that we can do. We can describe the patients, we can look into their survival, we can look into the progression and response from the disease, time to treatment discontinuation, the next line of treatment, and a lot more. So that is how we're trying and how we're enabling oncology research. And last and foremost, the most important part is terminologies. The current available standards and terminologies are not comprehensive and representative of everything that we need in oncology. So as a working group, we've been working on defining standards and the things that needs to be added. So for cancer diagnosis, we're using ICD-10, a set of cancer modifiers, and a new set of vocabularies called OMOP Genomic are being used to describe tumor attributes, anything else that we need to define the disease. For treatment regimen, we use HemOnc. Those of you who were at MedInfo, you probably heard a lot from Jeremy Warner about MedInfo and about HemOnc. And finally, we've developed new or de novo vocabularies that represent episodes of the disease. So all good. It's a lot of work, but we're not done yet. This is where we are today in terms of what we've done, what we've accomplished, vocabularies, and any toolings or any support that can help the ETLers and also studies. So some of them are almost at the end of so like you see like at the level of adoption or use in the studies in terms of the vocabularies, and some are still not. A lot of them are based on prioritization. What do we need and how should we actually move forward, where to start? There's also some work happening on like how can we support our ETL colleagues in terms of mapping genomic data. How can we define or detect regimen, and also providing like a lot of documentation for all the work that has been done, the work group has put together. And how are we doing it? So we've done a lot of work, and we've had to just do like a 360 review. And we realized that we have to focus to make it effective. It's a lot of work. It's just going to get worse, and everyone has their own interests. So it's a lot of prioritization of the work and the effort. We heard this morning about Patrick and how there's like a whole process for contribution and community support to support vocabulary. So the work group has worked together to plan a detailed community support for vocabulary, for oncology vocabularies. And I would like to invite you all who are interested to come and join us. This is still an open discussion, and this will only happen with your help and your contribution. We've implemented a real-time documentation for the ETL convention-based best practices. So as we develop, we put it on GitHub. And very importantly, we want to create an opportunity for broader engagement and collaboration. I'm very, very happy that Georgina is going to come after us and talk a little bit about some of the work that they're doing, how they're implementing it. Georgina and her group came to our tutorial 3 years ago. It was 3 years ago, and ever since we're just seeing more and more from this collaboration come out. It's really amazing to see that, and finally, everything is happening based on a use case approach. So again, open invitation. We need your use cases. We want to have them in, and we want your help to be able to prioritize and work on it. Come join us, and please, Georgina.
Thanks, Azia. So I've been asked to kind of join this session to give a bit of local flavour, talk about how we're using it in Sydney, and give a little bit of a perspective of how we're working with the clinicians and trying to help with really specific use cases for benefiting patients and patient care. So the project that I work on is called SAVAR, and that's for cancer variation. So the premise is all around building a robust methodology for understanding what is and isn't clinically justified in terms of variability in the delivery of clinical cancer care. But that's a collaboration between a number of universities, research institutions, and the public health system across the southern portions of Sydney. I'm going to share an example that may be familiar to some of the local crew here. I'm not really making sort of a broader comment on this except to pull out some really specific examples. But like a number of years ago, there was a bit of a scandal at one of our local hospitals where it turns out that there was a clinician who had kind of gone a bit off-piste and had decided that he didn't like the way that certain drugs affected his patients and had just sort of invented or figured out his own treatment protocols, and it went unnoticed for a really, really long time. But that's the background, but there's a few little pieces that I want to pull out and look at specifically here. So in the review of this instance, essentially the Chief Cancer Officer of New South Wales said, we don't actually know what effect that's had. There's no evidence for understanding what those dose variations really mean for the patient, so we can't really quantify it. Furthermore, so the clinician had a really strong belief that this was better, and it was based sort of on vibes, and definitely not on any material evidence there. But even in the face of sort of being called out on this and brought in front of the inquiry, stood by his own personal analysis because there is no meaningful way to have this evidence. Furthermore, he really firmly held those beliefs, and it was very hard to communicate with the clinicians, let alone the patients, what the effect of these dosing variations can look like. And that is pretty significant in the context of a cancer treatment protocol. We've talked about a few of the complexities of cancer treatment delivery, but specifically the one that I'm going to talk about now it's longitudinal. So the particular protocol that I've pulled up here, it's just an example from our local standards, which is EverQ. But it runs over the course of, I think it's 127 days, and 26 of those days require the patient to front up to outpatient cancer services to get IV therapeutics. So there's plenty of reasons. Purely scheduling alone, the burden of the treatment delivery itself is super high, super toxic. There are so many different reasons that treatment is warranted, patient preferences and things like that. So that's one of the reasons that some of these analyses become quite challenging. So our aim is to pull out measures of treatment variation that can fully account for that. And you can see these are all sort of the dimensions that we're considering and trying to look at. And we're trying to, the baseline in the literature is really focused around RDI, which is the amount of the agents that were delivered compared to as a percentage of the ideal dose. But that doesn't capture a whole host of things: delayed treatment, changes in schedule, individual dose modifications, rates of dose modifications, early termination of particular agents. So all of those ways of changing those doses, they do affect potentially the total dose in the same way, but they definitely don't affect the patient in the same way. They can have very, very different causes and effects and changes to outcomes. So this is we're taking a modularized approach to the implementation of the oncology module because it's still growing and it's kind of moving under our feet. And so this is the bit that we have leveraged to date, so this is going on the episodic nature that I think Azia mentioned. So looking at the diagnostic episode being kind of the parent, and then being able to ascribe the treatment back to the diagnosis that it's intended to treat. And when you're looking at primary and secondary cancers, that can be very powerful for being able to look at causality in the effects of those treatments. And then looking at the condition itself and fully describing that with the condition modifiers to be able to fully describe our patient population of interest as well. So just a couple of the examples of what this looks like in some of just simple examples from the data. And I wanted to mention on this slide, I did see one of the posters outside from one of our Korean counterparts having a very similar visualization for, people kind of start out all in line with the expected baseline of scheduling, and you can see as they progress through treatment, these effects of very small scheduling changes can become kind of cumulative, and they do affect the ways in which the doses are metabolized and the half-life of the drugs and the way that this will control the disease. And here's another example, which is looking more at kind of the combinatorial effect. When you're looking at different ways in which the patients can stop particular drugs and pick up other agents in practice as well. You can see that there's variation. Some patients drop the drugs a little bit earlier. A good example of this is here comparing the adjuvant and the metastatic setting, where quite similar baseline protocols will look quite different because they have very different intention. So the top one there is an adjuvant setting, whereby you're really trying to hit the, treat very short and sharp and hard, and you have the opportunity to potentially affect a cure in this patient. And anecdotally speaking with clinicians, they say, I'm much more likely to strongly encourage my patients to face up to these really bad toxicities because cure is on the table, essentially. But when you look at the metastatic setting, I think, it's accepted that chemotherapy is not going to be a cure for these patients, and the goal instead is to prolong life, manage symptoms, and control disease spread for as long as possible. And so instead of trying to hit it hard and sort of be one and done with your treatment, it's calibrating a dose that is tolerable for the longest period of time is essentially the aim there. And you can see that is quite evident from here. And I just wanted to call out that we're quite excited about seeing kind of Sally and her group here with all of their access to the much broader linked PBS data set, and looking at these kinds of things with agent combinations will be really powerful and open up a lot of collaboration in that space, I think. And just to end off, again, speaking to the right crowd here, as you mentioned, the HemOnc guys at MedInfo, because we had done a mapping between the Everq protocols and the HemOnc protocols, so Everq is our local chemotherapy or systemic treatment protocols, we already had in hand a mapping to the global standard and the one that's in the OMOP vocabularies. I had a bunch of conversations last week where we were already on the same page and able to communicate and have that harmonised assessments of those treatments. So yeah, those new studies are already in train because we were able to communicate via the harmonised protocols. So it's already been worth the effort for sure. That's all of my slides. Thanks.